Genetics is the science of heredity dealing with resemblance and differences within an organism. The father of genetics is a man called Gregor Mendel. He came up with two observations in his study of genetics. The first one is the law of segregation, which states that dominant and recessive traits are passed randomly to offspring. The second observation he made was the law of independent assortment, which states that traits are independently passed of other traits. And now we have the man, Gregor Mendel, the father of genetics. Dominant and recessive traits. Dominant genotypes only need one allele to show their dominant trait. Recessive genotypes have to have both alleles to show their trait. Big D and Big D would code for a homozygous dominant. Big D and little d would code for a heterozygous, and little d would code for a homozygous recessive genotype.
When two parents mate, Punnett squares can be used to determine the phenotype and genotype combinations. A basic example of Punnett squares would be here, where big R would represent red eyes, little r, white eyes. And when you cross a parent who's heterozygous with a parent who's homozygous recessive, and you do the Punnett square, the following will occur. A 50% chance of heterozygous, and a 50% chance of a child who's homozygous recessive, or 50% chance red eyes, 50% chance white eyes. And there are also different types of dominance, and one type of dominance is called incomplete dominance. In this example, big B would represent blue flowers, little b would represent yellow flowers. And when you cross a parent who has blue flowers with a parent who has yellow flowers, the following will occur. You'll have a 100% chance of big B, little b, or, in this case of incomplete dominance, a 100% chance of green flowers. A more complex AP-level topic on Punnett squares involve dihybrid crosses and sex-linked Punnett squares. So in this example, we'll be crossing two parents who are heterozygous for both traits, in this case, black color and rough texture. So, once you cross these, you're going to have to use the FOIL method to get the different types of combinations. The FOIL method involves first, outside, inside, last. So first would represent the first two alleles in the genotype combination. So in this case, it would be big B and big R. Outside would represent big B and little r. Inside, little b, big R, and last, little b, little r. And after you cross these two, these are going to be the genotype combinations. Lastly, we have sex-linked crosses. These are pretty basic to understand. So, when you have two X's, they would represent female. One X, one Y would represent male. Big D represents red hair, little d represents white hair. So when you cross a female who has red hair with a male who has white hair, the following will occur. You will have a 50% chance that a female with red hair who carries the gene for white hair, and then a 50% chance of a male who has red hair.
Thank you for watching our genetics presentation.